Hola, buenos días. ¿Se escucha bien, verdad? Bueno, bienvenidos a esta última sesión de las Santander Biomedical Lectures de esta temporada. Hoy voy a presentar en inglés al conferenciante. Para mí es un placer. Voy a decirlo. I will talk in Spanish. I'm not going to talk bad about you, okay?
I can hear.
Bueno, para mí es un placer presentar al doctor Paul Higgins, que es profesor de la Facultad de Medicina de la Escuela de Inmunología y de Higiene de la Universidad de Colonia. Professor Higgins obtained his bachelor degree with honors in microbiology in 1999. And he did his PhD in medical microbiology from the University of Edinburgh. His doctoral research focused on mechanisms on fluoroquinolone resistant in Acinetobacter species. Since 2015, he's a professor and lead the group researching dynamics of antimicrobial resistant mechanisms. He also heads the Molecular Typing and Outbreak Management Laboratory at the University of Cologne. His work bridges fundamental microbiology with practical hospital-based surveillance, outbreak investigation, and resistant monitoring. Professor Higgins' research interests span molecular epidemiology of the escape pathogen and Neisseria gonorrhea, Carbapenem-Resistant Acinetobacter, Klebsiella pneumoniae, and efflux-mediated antibiotic resistance and taxonomy of the genus Acinetobacter. In addition to his research leadership, he is a dedicated contributor to the broader scientific community. He serves as a guest associate editor for Frontiers in Medicine and Frontiers in Public Health, focusing on hospital infection caused by carbapenemase-producing organisms. He also is an active member of the ESMID and the British Society for Antimicrobial Chemotherapy. Professor Higgins has played a significant role in the organization of international scientific meetings, including the International Symposium of the Biology of Acinetobacter held in Cologne in 2013, Athens in 2015, and Coimbra in Portugal in 2023. He's a regular reviewer for numerous national and international research funding bodies, including the Wellcome Trust, the French National Research Agency, and the United Kingdom Health Protection Agency. Today, he has published over 116 scientific papers, and he has supervised over 20 PhD students. In his current role, he's at the forefront of genomic surveillance and outbreak response, sequencing carbapenem-resistant gram-negative bacteria, linezolid-resistant enterococci, and supporting infection control team in detecting and investigating suspected hospital outbreak. For me, it's truly an honor to welcome such a dedicated and accomplished scientist to share his insight with us today. So, Professor, whenever you like.
Thank you.
Okay.
Thank you very much. It's an honor and a pleasure to be here today and to give you this talk on Carbapenem-Resistant Acinetobacter Baumannii, the mechanisms and the molecular epidemiology. The perception for this type of work, particularly Acinetobacter Baumannii, is that it's very, very complicated. But today I'm going to try to give you a couple of different tracks. We're going to do carbapenem resistance, we're going to do molecular epidemiology, we're going to tie it together to the molecular epidemiology of Carbapenem-Resistant Acinetobacter Baumannii, and en realidad no es tan complicado. So, a little bit about Acinetobacter. There are 87 named species, they're gram-negatives, you all know that, I will not regurgitate this. You find them in a very diverse series of environments. By the way, if I'm talking too fast, wave your hand and I'll slow down. If you want these slides at the end, you could email me or email Alan, and you can have these slides if you want to look at them on your own. Anyway, Acinetobacter, you find them everywhere, but the most clinically relevant is Acinetobacter Baumannii, and particularly for things like ventilatory-associated pneumonias and a few other infections. and it causes outbreaks. It's intrinsically resistant to many antimicrobials, as seen here from UCAS with their expected resistant phenotypes. So the natural state of it is resistant to several different classes of antibiotics, including ampicillin, some cephalosporins. Here it says trimethoprim as well. So this is kind of what you'd expect. But it's not just the wild-type ones which are resistant, because we also get multi-drug resistant, extremely drug resistant, and pan-drug resistant. We found them in Germany. We did a clinical trial with Greece, Italy, and Spain. We also found pan-drug resistant Acinetobacter Baumannii there. You can isolate Baumannii from many different sources. So it's not just the genus you can find everywhere, also Acinetobacter Baumannii, from hospitalized patients, people in the community, from different types of animals, from the other environments, rivers, soil, and sewage. And one of the questions I'd like to, or hopefully you will be able to answer at the end of this, is it the same Acinetobacter Baumannii you find in this diverse series of environments, or are there particular niches that you find particular strains? Now, it's in the hospital that we find the MDR, XDR, and PDR strains, particularly in the intensive care units. And the reason why we have so much resistance there is we put down these antibiotic blankets, we have these bottlenecks where we're selecting for resistant organisms. But it's not just resistance which is being selected for. There are other things, host factors, resistance to disinfectants, ability to survive desiccation and to adhere to different surfaces. These are things we're also selecting for. It's not just resistance. And when we're looking at cross-resistance and the co-selection, this is a resistance island taken from an Acinetobacter Baumannii. It's maybe a little bit difficult to see here, but I'll talk you through it. These are all different antibiotic resistance genes. You have resistance to aminoglycosides, sulfonamides, rifampicin, beta-lactams, etc., etc. There's lots of different resistance genes that you can select for, but also for biocides and also heavy metals. So growth on mercury will also give you chloramphenicol resistance. Growth on disinfectants will select for beta-lactam resistance. And this is what we're facing, are these large resistance islands where you get cross-resistance coming in. The organism has a fairly large genome. I've looked it up. There's 48,000 genomes available in NCBI. Some of them seem a little bit low, 3,000 genes. Some of them are ridiculously high. Don't trust it if they have 7,000 genes. But with 48,000 of them, you could find the median. And they have about 4,000 genes that they have available in order to adapt to different environments. But they've also got these accessory genomes. They've got plasmids and other things here. And it's been estimated that there are 51,000 gene families available for Acinetobacter baumannii. So this is actually bigger than the human genome that they have out there as a pool of genes to pick up. Now, many of you are clinical microbiologists or work in diagnostics. You've seen this before. This is a multi-drug resistant, or actually a pan-drug resistant Acinetobacter baumannii, resistant to all these different drugs. And this is what it looks like. The impact of this, though, is much greater. This is taken from the Murray paper from a couple of years ago. It was a big review from 2019 data, and Acinetobacter baumannii is one of the top hospital-associated pathogens where you have death associated and attributed to antimicrobial resistance. And in fact, in Europe, there were almost 28,000 deaths in 2019 associated with Baumannii, 2/3 of which were carbapenem resistant. And because of this, the WHO has put Baumannii in the critical priority group of pathogens. There are many ways to become resistant. From these 6 different antibiotics here, we can have impermeability and efflux, we can have alterations of targets, we can have destruction of the antimicrobial, we can modify an antibiotic, and we can have additional targets. This is a topic of another talk. We're not going to go into depth in these ones here, but today we're going to talk more about what makes Acinetobacter baumannii carbapenem resistant. And for this, we're going to take a bit of a history tour. There's going to be 2 history tours today, one for resistance and one for epidemiology. The first carbapenem-resistant isolate came from Edinburgh in 1984. This predates the use of carbapenems, so there was already resistance out there before the drugs were introduced. The first report in the literature I could find is from 1991 in Germany, and they basically said It's resistant. We don't know why. We think it's impermeability. And impermeability was a mechanism which was often given for many, many years. So this is the non-beta-lactamase-mediated resistance. So it's altered penicillin-binding proteins, and it's for things like porin loss. And like I say, there's lots of papers on that. But to be perfectly frank, I have seen well over 3,000 carbapenem-resistant isolates, and this is never, ever a standalone mechanism. It does contribute to resistance. Impermeability is very important, but as the only mechanism, it doesn't exist really. Then we have efflux. Many people are looking for efflux-mediated resistance to carbapenems. And it's true, we can sometimes see very small increases of efflux, of MICs associated with efflux pumps, particularly with meropenem, but other times we don't. So really, efflux as a standalone mechanism, no. I think if anything, it's maybe strain dependent, and it gives a little bit of a nudge. So the MIC might be like here, then it could just creep it up a little bit. But as a standalone mechanism, again, no, this is not a good mechanism. And then we've got the enzymatic mechanisms. And this is the beta-lactamases. And we have different classes of beta-lactamase. We've got the class A's like GES and KPC, and we can sometimes find these things in Acinetobacter Baumannii. We have the class B metalloenzymes, things like NDM and VIM. And yes, we also find these sometimes in Acinetobacter Baumannii. Often it depends upon where the isolate comes from. North Africa, you find NDM. You find IMP, particularly in places like Japan. I'm not quite sure what you guys find here, but there are some cases of NDM in Spain. But be careful with looking for phenotypical tests for metalloenzymes. The literature is full of people that see metalloenzymes, but they don't really exist that commonly because the methods for phenotypical testing are very unreliable. And then we have the class D beta-lactamases, also called OXA, or the oxacillinases. Now, this is a very big group, and I will describe this to you all because the oxas can be very confusing. Acinetobacter baumannii has an intrinsic oxacillinase called OXA51, or OXA51-like. So we often use the word like after the number of the oxa because there are several variants there, and they're like them. So we have OXA51-like, and this is intrinsic to the organism. And then we have the acquired oxacillinases. These can be on plasmids and or on chromosomes. And this is OXA23 or OXA2440. We have the OXA58. We've got OXA143 and OXA235. And so these are the group of beta-lactamases that we find in Acinetobacter baumannii. This is some old work I did. This is looking at the DNA sequences of 484 different oxas. There are now 1,376 in the database, and it's ever expanding. It's easier to look at this figure than the bigger figure. And we can look at this figure. It's a little bit confusing. There's a lot of things going on here. But actually, we could break it down into three different parts. We can put a line here, and on one side, this is non-Acinetobacter. And on the other side, is Acinetobacter. And it's really nice how these things fit. The non-Acinetobacters are things you will have heard about. You will have heard about OXA48, a carbapenemase you find in E. coli and Klebsiella, and it's down here in green. And there are other oxas here you will know about, like OXA10 and OXA1. But also, some of these oxas are specific for particular species. OXA50 and 50-like you find in every Pseudomonas aeruginosa. There are oxas specific to Campylobacters and Achromobacters and other species as well. We're now going to look at this other part of it. We're going to start with the one on the left. These are the part of the Acinetobacter-associated oxas. And I have used a cutoff of 95% here, and everything in a different color is a different group. And each of these groups, but these are the acquired oxas. We've got OXA 23, 40, 143, 235, and 58. And these are the different variants that we find there. And as you see, many of them are very similar. The lines are quite flat. There's very, very small variation amongst them. And the other ones they are associated with different Acinetobacter species. Acinetobacter pittii has its own oxa, Acinetobacter caucaseticus has its own oxa, Acinetobacter baumannii has its own associated oxa. If you can sequence an oxa from an organism, you can, with a high degree of confidence, say what species it is. It's a very nice marker for these different organisms. Interestingly, Acinetobacter has two very close cousins. They have Acinetobacter nosocomialis and Seifertii. These do not have oxas. So there are a few exceptions to the rule that every Acinetobacter has an oxa, but most of them do. When we look at the OXA51 variants, this is the one which we find in Acinetobacter Baumannii, there is a lot of variation there. Often you will see written in the literature that OXA51 is a carbapenemase. It's not, with a few exceptions. Some of them are, some of them are not. And so you have to know which ones are which, and there's also other things have to be there as well, notably an insertion sequence called ISABA1. And we're going to continue this part of the story later on, or some of it. So this is a study from France, and they had a patient, had an Acinetobacter Baumannii with OXA66. OXA66 is one of the variants of OXA51. I know it's very confusing, all these numbers. But what happened was the MICs were raised at a later date. And this is because an insertion sequence called ISABA1 jumped in front of the OXA51. And this was a very good promoter. It leads to overexpression. You get a lot more oxa being produced. And they saw a 50-fold increase in expression, and the MICs were raised. But for most of you, will probably be looking at and think, that's not very impressive. The MIC's gone from 2 to 4 and 3 to 6. That's not very good. We saw something very similar happening in patients. And we also saw ISABA1 jumping in front of this intrinsic gene. We also saw an increased expression. We saw 46, the French saw 50. It's the same. But what happened here was there was a mutation in the OXA66 gene that changed one amino acid, leucine 167, into a valine, and this is what gave the much higher MIC. So now we go from 2 and 4 to more than 32. So we have this leucine to valine substitution, and we have ISABA1, and these two things together give you the resistance. And that basically is Acinetobacter Baumannii carbapenem resistance. You need to have overexpression, and you need to have the right variant. So we're leaving carbapenemases right now, and we're going to bring the other track in. We're going to look at the molecular epidemiology. And this is basically what I do for my day job. The hospital phones me up and says, there are 2 patients with MRSA. Is it the same strain? We've got a cluster of Klebsiella pneumonia on the baby ward. Is it the same strain, etc.? So this is what I do for my living, molecular epidemiology. And we're trying to answer simple questions. Is strain A the same as strain B? How close is strain A to strain B? The typing nomenclature for Acinetobacter baumannii is a little bit complicated. There have been lots of different ways people have described the organism. We've got sporadic and outbreak, European clone, sequence groups, and a whole bunch of other ones. To make life easy, we just call them international clones, and we have a definition. This definition is old, perhaps it's a bit weak now, but basically if we see 2 isolates or isolates from different countries that are very close together, there seems to be some sort of international dimension to this strain. It's an international clone. And this whole story begins in 1994 with work from Leni Dijkshorn and others from Leiden in the Netherlands that were using some sophisticated methods back then, AFLP, and looking at outbreak and sporadic strains. And they had them from various European countries. They basically found that outbreak strains were more resistant than sporadic strains. And basically the outbreak strains, they clustered into 2 groups. And they're all from Europe, and they called them European clones 1 and European clone 2. So that's quite straightforward. 8 years later, they did the same work, and they found a 3rd clone. And all these isolates were gentamicin resistant, so it's like one bottleneck. There was ciprofloxacin resistance, another bottleneck, and a third thing was beginning to emerge here, carbapenem resistance. So this is like a third bottleneck. So remember, all of these things we're constantly selecting for organisms. We're constantly selecting. So here we've got at least 3 different bottlenecks. This was using AFLP, and very few people used AFLP. But in 2005, a portable method came out, MLST. Everybody know what MLST is? Multi-locus sequence typing. It's based on 7 partial genes that you sequence and you compare them, and it's a way to group things together. If you're doing typing in the hospital, it doesn't have a lot of resolution, but you're able to group things together and say, these are quite similar and these are quite similar. There was a second typing scheme came out for MLST a few years later, and this is where the first term international clone came out. Round about the same time, in Cologne, we were using a method called RET-PCR with the DiversaLab system, and we had got hold of about 500 carbapenem-resistant isolates from all around the world, and we typed them, and we found 8 clusters. So our first 3 clusters were the same as clones 1, 2, and 3 from the European clones, but we found these 5 other clones there. And when we look at these as an international clone, what's interesting is that, well, first of all, very few of these isolates were singletons. Most of them clustered with one of these 8 clones, so we've got 43 isolates from 28 centers that did not cluster with these 8 clones. And these other ones, they're more intercontinental. Clone 1 and clone 2 we find in Europe, in Australia, in South Africa, in the Middle East, the Far East, Asia, and the USA. So they're truly intercontinental, not just international. But interestingly, our clone 5 was particularly rich in Central and South America. We have like 1 isolate there from Spain and a couple from Germany, maybe tourists and things like that. But Argentina, Colombia, Mexico, Venezuela, the USA, this was particularly rich for this clone 5. So up until this time, we have 8 different lineages of Acinetobacter Baumannii that are carbapenem resistant, and very few, less than 10%, would be not one of these clones. That was then. Now we're in the genomic era, and we no longer use RET-PCR. MLST, 7 bits of genes. But instead of that, we now got the whole genome. We can now compare thousands of genes into what's called core genome MLST, and it gives us a much higher resolution. And so we end up looking at actually 2,390 genes. This is what we call the core. It's something that all the organisms, all the isolates of the species should share. So we've got something that we can compare. And we can use it for typing, for outbreak analysis, but we could also use it for wider analyses looking at the global populations. Now, jelly beans come in 50 flavors, and Heinz is famous for its 57 flavors, but Acinetobacter Baumannii comes in hundreds of flavors. Each genome has its own unique flavor or signature. I like flavors. We've now found 11 international clones, and actually we're beginning to see many, many more of them kind of coming up. It's almost every day we're seeing, okay, these isolates are clustering together, these ones are clustering together. So at some point, we have to up the numbers of international clones. And this is what it looks like. We had a follow-up survey from one from 2002 to 2006. We've got one from 2012 to 2016. This time we have 313 isolates from 47 countries, 114 study centers. So we've got a very wide spread. And when we sequence them and put them together into this tree, we can see our different international clones. We've got IC2 in the middle, international clone 2, IC1 at the bottom left here, international clone 1. We've got clone 6, clone 4, clone 8, etc., etc. There's one clone just from the USA. It's not international. Americans are not very international. They're very they like to stick to themselves. They have their own clone. Make Acinetobacter great again. So, and the distances between these clonal lineages, they're different in about 2,000 genes. We're looking at 2,390 genes, and if you compare one clone to the other, they differ in almost 2,000 clones. This is like 2/3 of the genome. Is it okay? Yeah? All right. So they're very different. And so basically what we have is it's a bit like the tepuis in Venezuela. Anybody know the tepuis in Venezuela? You got these mountains, they go up, it's like Roraima, they're very flat topped, and then they go down to the valley, and then 2 kilometers away, there's another one, there's Kukanan. And so this is what the clonality is like. You've got these big clusters and a big gap to the next one. Within one of these lineages, you've also got variation. 400, 500 alleles are different. But still, from this lineage to another one, more than 2/3 of the genome is different. So now we've got 11 international clones. The kind of the view that we saw from 2002 to 2006 is very similar to what we saw in this study here with the genomics. We saw that international clone 2 was the most prevalent clone. We also see that clone 5 is again particularly rich in Central and South America. We still don't know why. We don't know why this is the case, but it seems to be like that. In fact, international clone 2 is the one which causes the predominant outbreaks around the world. Wherever you go, you have an outbreak, it's nearly always going to be international clone 2. Here are some examples that we've done. We've worked on in North Africa, the Middle East, in Brazil, in Afghanistan, in a German hospital, in Sudan. It's the same case wherever we look at. International clone 2 is the clone that you will find most often. Again, South America, they have their own thing going on there. They have clone 2, but they also have a lot of clone 4, 5, and 7. For some reason, Argentina does a lot of work in Acinetobacter, so there is a bit of a publication bias from the bottom of South America. But you also find in Bolivia, clone 7 is particularly rich as well. So wherever we look, we see this slight difference. Going to go back to this OXA51 again, because remember I was speaking about genomic flavors and things like that, and each of these lineages are associated with a particular OXA51. So if you're an international clone 1, you've probably got OXA69. If you're international clone 2, you've probably got OXA66. If I see an isolate and I sequence it and I see OXA71, I think it's probably a clone 3. So actually, we can begin to look at these genomic flavors to help us when we're trying to look at the molecular epidemiology. And this is also what we see when we have overexpression of the intrinsic OXA. Remember I said how we saw this leucine to valine at position 167? We see this happening quite a few times. And because we know the clonality, we know that OXA66 becomes OXA82 because those isolates cluster together. We know that OXA69 becomes OXA107, and they all mutate at the same position, and they all have ISABA1, and this is how they become resistant. So you need to have the right variant with ISABA1. When we did our first study, the international one, and we looked at why are they resistant, the overexpression of the intrinsic OXA was the most common mechanism. So 193 out of 490 isolates had that, followed by OXA23. Our more recent survey, now remember the clonality between the first and the second was more or less the same. There wasn't much differences going on there. We saw the same guys with the same ratios and the 10-year difference. But by 2012 to 2016, OXA23 was by far and away the most common mechanism. So 234 out of the 313 isolates had that. And the overexpressed intrinsic OXA had completely plummeted. So the clonality of the isolates has remained the same, but their mechanism of resistance has now shifted. No longer do they overexpress the intrinsic OXA, They're much more likely now to have OXO23. In fact, OXO23 is by far and away the most prevalent one wherever we look around the world in outbreaks. We had this survey in Greece, Italy, and Spain. It was more than 80% of isolates. There are plenty of publications out there from Germany, Greece, and China which said, we used to have a lot of OXO58, now it's all OXO23. Spain and Portugal were famous for OXO40. It has still here, but it was being overtaken a lot by OXO23. Now, we're in Spain, so I had a quick look. There are 712 papers on Acinetobacter Baumannii. I looked at some of the more recent ones to see what they're saying as well. This is a nice paper from Lucia here, which this is an OXO40 outbreak. And what I like about this one here is this area, because here we've got isolates from 2009 and also 2020. They're the same. We're not seeing any differences. We always think that bacteria are constantly mutating. It's not always the case. Some of them are very stable. I don't know where this isolate has been hiding in these years. Maybe it was in the fridge somewhere and someone brought it out again to infect people. But anyway, OXO40, this is what we sometimes see in Spain. We also see OXO23 and NDM from Spain, and NDM6, this is clone 9. Also, the first ones here is clone 2 and clone 3, ones that you'd expect to find. This is from Andalusia. They're seeing mostly OXO23 and mostly international clone 2, and these isolates are spreading throughout multiple units. So basically, what we see around the world, we also see happening here. A lot of OXO23, a lot of clone 2. What are the alternatives to carbapenems? So all these isolates are resistant to carbapenems. So we've taken our 313 isolates from the ones that we got from 2012 to 2016, and we have looked at them against other antimicrobials. So tigecycline is often given as an alternative. It has very poor serum distribution, and when we looked at our isolates, more than half of them were resistant to tigecycline. We don't know what therapy these people have been having, but more than half our isolates were resistant to tigecycline, and it was all mediated by efflux pumps. There's colistin. We know people want to use colistin, has a lot of renal toxicity. From our isolates, there was very low resistance rates, less than 5%. But resistance to colistin in Acinetobacter comes through mutations in this PMR-CAB operon. And when we were doing our Magic Bullets clinical trial, which was comparing empirical use of meropenem and levofloxacin against colistin and levofloxacin, when we started, there was no colistin resistance in Greece, Italy, and Spain. But when we started this clinical trial, then we saw colistin resistance. So basically, if you use colistin, you will have resistance, and it will develop very quickly. So colistin, I don't know if you use colistin, but if you're using it for Acinetobacter, resistance will develop very quickly. It's not a very good drug. Cefiderocol, which is a more recent antimicrobial. It's a siderophore cephalosporin. It hijacks the siderophores, gets pulled in, and has activity. We actually saw that most of our isolates were susceptible to cefiderocol. We did have a few which were resistant. About 57 of them were resistant to cefiderocol. The resistance mechanism is a little bit hazy. We don't quite know. It could be due to NDM or GES, these metalloenzymes, but also some ESBLs like VEB and PER can cause resistance. There are some altered penicillin binding proteins which are associated with resistance, but we have isolates with these mutated penicillin binding proteins that are susceptible to cefiderocol. So the jury is out how you get resistance, but this seems to be a fairly good alternative. And then sulbactam, darlobactam. We actually saw that, so sulbactam has an antimicrobial activity against Acinetobacter anyway. Darlobactam has even more, and we can see down here from this figure that when we give sulbactam, darlobactam, compared to the sulbactam, there are 66, 97, 56, and 7 isolates with these low MICs versus just 2 with this MIC there. Interestingly, the addition of imipenem to this has no effect. Imipenem really didn't do very much when you put it to the Sulbactam, Durlobactam. So there are some alternatives out there. This is just an added extra. This is the ISABA1, this insertion sequence, because everybody is always looking for a resistance gene that causes resistance. But actually with Acinetobacter baumannii, all you need is this insertion sequence. It is a transposase with a little bit of DNA at either end of it. It jumps into the genome in different positions. It will disrupt, and it will lead to overexpression. And we have all of these different antimicrobials that you will get resistance to just with ISABA1 coming in with the chromosome. So you don't need to have a large array of resistance genes to become resistant. So how am I doing for time? Okay. So up until now, all of this work has a publication bias because we're only looking for carbapenem-resistant isolates. So one day I was speaking to my colleague Harold Seifert, and I said, we have a publication bias. We have to address this. So we decided to take all the Acinetobacters that come into our clinic, into our diagnostic laboratory, and keep them. So we started this 5 years ago. We have now collected 250 Acinetobacter Baumannii, and we have sequenced 192 of them. So most of these isolates are not carbapenem resistant. So now I can do a comparison of carbapenem resistant to non-carbapenem resistant, just to see if we're seeing the same epidemiological situation. So this is a different type of tree based on core genome MLST. It's easier to have a look at here, but I'll just talk you through this one. Basically, the distance from this guy to this guy is about 2,000 genes. It's the same as we saw with the earlier tree, just a different way of visualizing it. And what we have here are little clusters at the end of these branches, and these are the international clones, clone 7, 11, 6, 1, 2, and 9. And all these other ones here in blue, these are not international clones. And most of these are at the end of a line. So this would be about the 2,000 genes different to the next one, to the next one, to the next one. There's a few things here which look like clusters. They're not really that close. Sometimes we have a patient that will come back and come back and come back and come back, and each time we have a strain, and there might be a little bit of differences going on there. But we don't really have any outbreaks of these non-lineages. We have 55 international clones and 137 singletons. When we look for carbapenem resistance, those that are carbapenem resistant are the international clones. It's the same guys, except one. So only one of our carbapenem resistant isolates was not an international clone. Quite similar to what we see in our worldwide surveys. So if you are carbapenem resistant, you are more than likely to be one of these small, narrow numbers of clones. Also interestingly, ISABA1 is a very good marker for multi-drug resistance because those with the ISABA1 in blue are mostly the international clones. There are a few isolates here which have ISABA1, but what's interesting is they have one copy. And these other guys, they've got like 25 copies. So if you're able to quantify within a strain, do you have ISABA1? And the answer is no, you're probably a singleton. If the answer is yes and you have multiple copies, you are probably going to be a carbapenem-resistant international clone. So we've now seen carbapenem-resistant isolates. We've seen non-carbapenem-resistant isolates. We've seen the clonality there. If you're carbapenem-resistant, you're one of the clones, and if you're not going to be one of the clones. What about from other sources? At the beginning, I said you can find Acinetobacter from all different sources, including animals. So we're going to spend 5 minutes or so looking at Acinetobacter from animals. And when we're talking about animals, we have to be careful because not all animals are created equal. We have got companion animals, dogs and cats. We've got livestock, chickens, cows, sheep, and we've got wild animals like mice and birds. And this is important because there are some people that are, I would say, are misinterpreting the literature, and they're producing these high-impact papers looking at Acinetobacter from non-human sources, and is it going to be a big problem? And they produce these heat maps with very impressive looking array of antimicrobial resistance genes. But remember, the organism is intrinsically resistant anyway. So many of these things, it's default. It's a default set of genes that they have. There is nothing that makes them special. And you can chop all that down there and say, okay, most of these are actually irrelevant. There's like one here from a sheep that we'll talk about in a minute that is really quite a relevant one. But in this case, most of these genes are actually intrinsic. So don't be impressed by them. So from companion animals, dogs and cats, you can find, this is from Germany, they found carbapenem-resistant isolates in dogs and cats. They had OXA23, they had overexpression of the intrinsic OXA. They saw lots of resistance, some of which is intrinsic, some of which is acquired. They did some pulse fields. They found out that these isolates were international clone 1 and international clone 2. And just like human strains, they were also spreading between clinics in the veterinary world. So there's a bit like a hospital-associated thing going on there. Looking at wild animals, really they're not finding much antimicrobial resistance. This is looking at storks. Sometimes from chicken and geese, livestock, you might see a little bit of resistance to things like aminoglycosides and sometimes also tetracyclines. Interestingly, when you're looking at wild birds and storks, these things migrate from Africa. They go up to Europe, up to Poland and Germany. They don't have Acinetobacter to begin with. It seems to be seasonal. It comes in springtime into summer, then they fly away. So they're not always colonized with Acinetobacter Baumannii. And when we look at the clonality of these isolates, most of them are on their own. They're like those singletons. The avian wildlife isolates do not cluster with human ones. And you sometimes see a very small link to some of the clonal lineages with livestock. Livestock might be a slightly more important source than wild animals. This is from cattle in Germany. They found Acinetobacter Baumannii in the nose of these cows. Particularly, they saw some of these isolates were clustering with international clones. Again, there seemed to be a seasonality to it, that in the springtime you don't see much, and later on you see more. They saw resistance, but most of this is expected resistance. There wasn't much unexpected resistance going on there. And they actually linked this to the use of sewage sludge as a fertilizer. And looking at other food animals like turkeys and chicken, you can sometimes find some resistance genes in there. But with some of these papers, their species identification are maybe not entirely good. So they're seeing isolates there without OXA51. OXA51 should be found in Baumannii. If you don't see OXA51, it's not a Baumannii. So you've got to take some of these data with a pinch of salt. And then this one here, I think, is the most interesting. There was in these sheep from Pakistan where the animals, about a third of this flock of 1,200 animals, died within 3 days of symptoms. They identified an Acinetobacter Baumannii. They tested Koch's postulates. They took the infected animal, they cultured the organism, they put it back into a sheep, and it died. And this was a clone 2, carbapenem-resistant. So rather than it being a zoonotic organism, it seemed to be anthropic. We are the ones that are producing the resistant isolates which are going to kill the sheep. And there are plenty of other studies looking at cats, dogs, horses. Depending, if they're wild animals, you don't often see much resistance. In where their vets has been using antimicrobials, you will have selection. You will begin to see the resistance developing. We can put all these isolates together into one tree, and this is what I've done here. And what we see from this is international clone 2 clinical isolates. This is from sewage wastewater in Croatia, which is the same isolates as you find in the clinics. We've got one cow isolate here, none down here. And in this part of the tree, the vast majority of cows and bird isolates are at the end of these branches. They're all on their own, with one or two where we see a very, very far away link to this isolate here, which is from a bird, or these ones here. But they're not that close. They're really quite far apart, but they're slightly associated with it. So, in summary, because we're at the end now. There are basically two types of Acinetobacter Baumannii. We've got the wild types. They're singletons. They're not part of the international clones. They don't really have ISABA1. They're intrinsically resistant to some antimicrobials, but they're just the default. And then we've got the carbapenem-resistant isolates. They're international clones. They cause outbreaks. They're expanding all the time, this clonal expansion of these things. You find them everywhere, and they always have multiple copies of this ISABA1. So, the molecular epidemiology of Carbapenem-resistant Acinetobacter Baumannii, resistance is mediated primarily through oxacillinases, particularly OXA23. ISABA1 is often involved. I didn't mention it, but ISABA1 is necessary for OXA23 as well. The OXAs are species-specific. You see an OXA 500, you can look it up and you figure out what species you have. You have OXA 282, you know the species. The intrinsic OXA of Acinetobacter Baumannii is very helpful. It tells us what it is, it's the Baumannii, but also it can help us to understand if it's one of these international lineages as well. The genomes have different flavors, but some flavors are more widespread and more common and more popular than others. Where all Baumannii isolates have the potential to become carbapenem resistant, what we do not see with the non-lineages are them spreading through people and causing outbreaks in hospitals. So they're probably just a singleton that might be you see once in one place and never again. And basically that most of the carbapenem-resistant isolates belong to a small number of international clonal lineages, and Baumannii does not respect borders. This is the work never of one person. It's a huge team of people back in Cologne and people that I've worked with, including Lucia here in the audience, that have been very good help and collaborators with me over the years. I want to thank you for your time, for coming here to hear me talk, and I'd be happy to take questions. Thank you.
Thank you very much for this nice
45 minutes.
Okay, so if you have any questions.
Paul, thank you very much. It has been a very, very, very good lecture, very interesting, and congratulations for your work.
Thank
It's
you.
fantastic. I have a little question. Have you seen any differences between, in clinical strains, between colonizing strains or true strains with a patient with clinical disease, infectious disease?
What's the first? Sorry.
Yes, in clinical strains,
Yeah.
we can find two types. Only strains that colonize
Okay, yeah.
the patient, or you say that strains truly infect the patient. Have you seen any difference between that?
That is a very good question, and it's one of those things which I can't answer, because I'm not a clinician, but I used to work with a clinician for 2 decades, Harold Seifert, and we would often deliberate what is a true infection or not.
Mm.
I think when we're looking at those from a lot of our isolates we get in the hospital are from surveillance. And I sometimes see there are some from bloodstream infections. So from our isolates, we have very few bloodstream infections. We've got like 3 or 4 Baumannii from bloodstream infections. Most of them are surveillance swabs, rectal swabs. Some of them are from wound swabs. Some of them are from bronchoalveolar lavage. But I don't actually have it broken down into the different ones there. That's a good question. We actually, when we are collecting all the Acinetobacters, we've got all the Baumannii, but also we have a lot of other species as well. We did see a difference with, I think it was Acinetobacter pittii. We saw more urinary tract isolates with pittii than with the other ones, but I'd have to check that data again. But it's a very good point. How many of them are true infections and how many of them are just colonizing?
Yes, because if you, for example, if you know that that the strain is from blood culture
Yeah.
or other sterile normal fluids, it could be a very good point to see if that strains are different or not in relation with the others. It could be a good point to see, I think.
Yeah, I think it's also an antibiotic stewardship question because in Cologne now, they no longer give out antibiotics just because you find urine and bacteria. They no longer have like 10 to the 5 CFUs per mil or anything like that. They basically only treat people if they have symptoms. So this is a very good point, is that infection or colonization. But this is something I will have to go away and think about. Thank you.
Thank you, thank you.
Other questions? Well, I have a question regarding to sequencing and bioinformatic analysis, and I have a doubt with the international clones, especially the IC2, the international clone 2, because you have a very different spectrum of strains. Do you think that when the scheme for the core genome MLST is correct, or it should be changed? Regarding, because in one of your slides shares that you have very different amounts of genes, especially accessory genes. How do you deal with that when you are grouping all the?
I mean, the thing about accessory genes is you can use them for typing, but you end up in a situation. So we had a bloodstream, we had a patient with a bloodstream infection with Enterococcus faecium, and from one of the blood cultures, you got 2 blood culture bottles, one had a VRE, one had a VSE. So they're different, but they're actually the same. It's the same strain. One of them had a plasmid with the vancomycin and a couple of other resistance genes, and the other one didn't. That was the only difference between them. So at the core genome, they're the same, and the accessory, they're different. So if you were, and this is a problem sometimes using accessory genomes, because they are accessories, they can discard them, they can pick them up. It's not something which you would necessarily, I would not put a lot of weight onto some of these accessory genes. They're interesting to be able to differentiate between them, but ultimately you have this situation where it's the same strain, but it's different strains. We have a Baumannii, which was infected. It came into our hospital from a patient, had NDM and OXA23. And at some point, this isolate moved to a different part of the hospital. Basically, the patient went to a dialysis machine. 3 weeks later, somebody had dialysis, and they picked up the Acinetobacter from that room and took it to their ward. And then this isolate moved around. And from this cluster, most of them had NDM and OXA23, but one of them only had OXA23 from the same patient. So phenotypically, they're more or less the same. They had the same antibiogram. Genotypically, if you're looking at the accessory genome, they were different, but actually it was the same strain. So I tend not to use the accessory genome too much because it can lead you to false conclusions.
So it's not good to use the whole genome?
It depends on the question.
Yeah.
I think it depends on the question. If you're looking for outbreaks, then you could link it. But again, these genes, many of these genes are acquired and lost. And within the patient, you can have both forms at the same time. And they could then spread, but I find that it's not too helpful because you end up saying, okay, they're different strains, there's no transmission, but clearly it's the same strain. So accessory genome is useful, but I don't put much weight in it for typing. But maybe I asked different questions.
Okay. Okay. Any other question? No more questions? Well, I have another question.
Okay.
Apart from the resistance with Acinetobacter, what do you think is the success of this pathogen?
Or what makes it successful?
What makes it successful? I mean, in terms of pathogenic.
I mean, one thing is its resistance to antimicrobials, but it's not the main thing. I think it's transmissibility and longevity. So you can typically, if you were to have an E. coli and put it here, and an Acinetobacter and put it there, and come back in a week, you probably get the E. coli and the Baumannii, but come back in 2 months, you will not get the E. coli. The Baumannii will still be there. And this, I think, is what is one of its main virulence factors, is its ability to survive and to be picked up again and to transmit to someone else. I think also there's a lack of competition. So it's a bit like if you have a forest and you chop it down, or you have a field and you take away the wheat, then these weeds grow up. So the organism doesn't have a lot of competition, which is why it can colonize people quite easily. I have an E. coli in my gut, you have an E. coli in your gut. If I give you my E. coli, it's not going to be found in your gut. You have your own E. coli, it's going to kick it out. If I have an Acinetobacter Baumannii in me and I fling it onto you, it will colonize you. So it has the ability to colonize fresh hosts, and I think this is part of it. We see these lineages all around the world, so they have something in common. And we see these non-lineages also becoming multi-drug resistant. So multi-drug resistance is a feature of the organism, but it's the ability to transmit and persist is special in some of these lineages. So, but we don't know why that is. You know, what makes clone 2 successful compared to these singletons, nobody has yet been able to address. People have looked at biofilm, but they all produce biofilm. People have looked at desiccation tolerance. They're all pretty desiccation tolerant, so there's something else there. It might be disinfectants. I'm not quite sure, but there is definitely something out there. Okay.
Regarding these resistant categories, do you think they have to change? Because now with the new antibiotics and combinations, when you talk about the pan-drug resistance, it is really a pan-drug resistance strain. How can we define a pan-drug resistance strain?
Yeah, I mean, if you go back to 1992, multi-drug resistant isolates, would you still call them multi-drug resistance now? Because now we have newer drugs which they have to be resistant to. So yeah, definitions change with new drugs. So at some point, you will have to reapply these rules. So everybody's citing a paper from, is it Margolis, from 2015 or 16. They have a paper which is where they define MDR, XDR, and PDR. But since then, other drugs have come out. So yeah, we have to redefine pan-drug resistance. So our pan-drug Baumannii from Germany, it's probably not pan-drug anymore because cefiderocol is out, or sulbactam, darlobactam. So yeah, definitions have to change with the times. Good point. No, thank you. And remember, they can have the slides if they want. If they want the slides, they can have the slides. If they want the slides. Yeah. Is that okay?